Okay, so it's Monday afternoon, the last Monday in August. Um, Next week will be Memorial Day. I'm sorry, will be Labor Day. And the summer will be over. It's kind of hard to believe our second summer living through COVID. Um, Michael's going to be joining me in a few minutes. But what we wanted to talk about today, um, besides, I want to hear if anyone um, um, reached out and did remarkable things. Um, um, you know, doing something outside the box to change the world that we live in. So we want to hear those stories. But um, um, what we're going to talk about today, or I'm going to talk about, and Michael will join me when he gets here, is I want to talk about drug development and what that means, what drug development means. And here's Michael. Um, Michael, you there?
Absolutely.
Great. So I was just teeing up our conversation about what drug development means for us as consumers, how it influences what options we get in terms of supplements, in terms of drugs, in terms of treatments, how those treatments get labeled, and the such like. And we wanted to talk about um, that for our topic today.
It's a great topic, isn't it? Because it brings back to actually consumer. Uh, it, it sits on the consumer actually to really try to find out um, what are the influencers that bring these drugs or supplements to market, and what are the influences that affect the messaging. And you know as well as I do that there are a lot of pressures um, to bring new supplements and drugs to market, and it's all about really being able to own the intellectual property and be able to make profit. And I think that comes back to some of the tension that we talked about earlier today, Joe, and that is that you notice that pharmaceutical companies are trying to patent natural molecules like N-acetylcysteine or natural molecules like um, red yeast rice. I mean, it's red rice yeast. It's all they're trying to patent naturally occurring molecules, and if they don't aren't if they're not able to patent, then they don't. There's not a return on investment to study them, and thus becomes the grand disparity between so many of the things that we find in naturopathic medicine and also in nature and also in diet and lifestyle that are really healthy for us, but it doesn't get as much press and it doesn't get into journal articles because it's not funded by another business.
Yeah. Well, the piece that's interesting there is what you said about intellectual property. And what do we mean by that? Intellectual property is the ability to patent a product and make it yours. And for you to be able to do that, two things have to be the case. One is it has to be something that's novel. And number two, there really has to be nothing that that suggests that nothing called prior art. So nothing that was there saying, hey, here's a paper that gave us a good idea that curcumin works for inflammation. So you did a study using curcumin to treat joint pain and it worked. Well, unfortunately, there was prior art and it wasn't novel. The one other way that you can get a patent besides having a novel thought, is if you have a novel thought about method. So if there's a natural product like 8-parental nerinogen, which is the most potent phytoestrogen that there is, which is an ingredient in hops, it represents a very small fraction of what's there with the xanthomyol, which is one of the components of hops. Essentially, when you take out the resins and you take out the isoalpha acids, some of the oil-like compounds that are left, are xanthomyol, and 8-parental nerinogens there, and it's in a very small amount. Well, that isolation is fairly tough. If you came up with a method to chemically synthesize it, or if you came up with a method to isolate it better, that would be a methods patent. And you'd be able to get some traction around that.
And then correct me if I'm wrong, because you've had a foot in many different arenas, and one of the arenas that you've had your foot in is in nutrition research for different companies that have a method and identify a molecule that has that, and the extraction or the method to make it bioavailable or available in a new form for the consumer is really the method patent that allows so many of these things from nature to move forward.
Right. Yeah, I was the medical director briefly for Kindex Therapeutics before it became Kindex Pharmaceuticals, which was an attempt by Metagenics and Alticor to spin off a company knowing what we knew about the isoalpha acids in hops, and seeing if we had the potential for a drug and a potential for a drug that could treat either diabetes or possibly PCOS. And we didn't just have method, we had science that was novel, because number one, we learned some things about the structure that had not been previously known, and we had some research that demonstrated its ability in those fields. And the difficulty is some of the hurdles that are put in place. The class of diabetic drugs, the thiazolidinediones, which ran into cardiovascular side effects in terms of congestive heart failure, led to a whole category of cardiovascular safety studies that had to be done for diabetes drugs. And coming in with a novel botanical ingredient where we weren't 100% sure of the mechanism, didn't make anyone at the FDA, after the cardiovascular complications of, the COX-2 inhibitors and the thiazolidinedione drugs and heart failure and the need for cardiovascular safety. It put such a high hurdle for what we really have to do on safety work that it became a barrier to getting there. Interestingly, the workaround for a lot of these things is to come up with a new chemical entity. So you basically take something like resveratrol, shown to affect the sirtuin genes, and you put it into a test tube and you play organic chemist for a while and see how many stable compounds you can come up with that are chemical modifications. So they're new chemical entities, they're new to nature variants on the resveratrol, and then you take them down the drug development path. That model was a lot more attractive a few years back when David Sinclair was doing his work on the sirtuin genes and resveratrol, and a company that he was doing research for, and he was a part owner of, called Sirtris, sold itself to what is it? EL Lilly? No,
EL Lilly.
EL Lilly sold themselves to Lilly for $720 million because they had about 5 new chemical entities in the development path, and we've never seen any of those, which speaks to some of the risk involved here.
Yeah, certainly. The other, well, we could go on and on with examples of, well, it's what pharmaceutical researchers do. They
Right.
go to the areas of ethnobiologic diversity, see how different plants were used therapeutically in the population, and then basically try to isolate the molecule that has the greatest biologic effect for the more and more specific condition.
Right.
And thus that interesting interface, isn't it, between ethnobiologists, pharmaceutical companies, and drug sales in the United States,
Yeah.
right? Or the world.
Hence, there are companies now developing new chemical entities around psilocybin, for example, as a potential growth industry, because the psilocybin has so much prior art that other than method, patents aren't going to be available. But I think it's really important to point out to people, you were just talking about the libraries of botanicals. It's really important to point out how this is done. Basically, the libraries don't just include plants, they include like every compound that you can think of. And and a lot of the old drugs and a lot of the modifications of the old drugs. And someone comes along and says, hey, we want to treat such and such a disease. And we know that the target for that in a cell is a certain biochemical pathway, right?
Yeah.
So when Roger Newton was doing his work with looking for a better statin than Mevacor, which was the original statin drug, he went looking and they selected a whole series of chemically modified statins, and they picked one out and they saw that it was active on that pathway. But they also screen for like all sorts of other compounds which are going to inhibit HMG-CoA reductase. And it leads to an interesting narrowing of focus, right? Because we said heart disease is about cholesterol. We'll talk about HMG-CoA reductase. And it took a long time to realize that the whole category of statin drugs also had anti-inflammatory properties, because they weren't in that initial screen, because people just screened for HMG-CoA reductase inhibitors.
That's exactly true. And then when you look at the structure of most of statins, is you realize their structure is very similar to the structure of vitamin D. And so the question is, are statins having a mild vitamin D effect, or is vitamin D having well, we know it has an anti-inflammatory effect. Does vitamin D have a mild, quote unquote, statin effect? And that's the crossover and the fascination of molecular structure, vibration of different chemical bonds, and the interaction with infinitesimally small changes in our biochemistry.
Right. And it leads to that naming bias of how things get labeled. Like this is a cholesterol drug, because once we've done something like identified that this specific drug hits this target, a lot of times we don't even know if that's safe. So the first thing that's done is you take whopping doses of it and you give it to animals as single doses. And if they survive, then you give it to a group of animals at multiple doses. And if they survive, then you say, okay, is there a model for this? Like, is there a model for atherosclerosis and heart disease? Yep, there's a model. There's a group of mice which are particularly good for studying this in. Then you start giving them the tolerated doses and look to see if you have an effect in the mouse. And if you've passed that level, you finally get permission from the FDA after having done a couple different species and having demonstrated that it seems to be safe in levels that would be the human equivalents. You then go into humans and you give the first dose to humans in a concentrated dose, and you basically look to see if any of them die. I mean, it's harsh to say this and it's brutal to say this, and you've done a lot of diligence to say they shouldn't, but you know, it's actually like in the first study, you don't give on day one of the study, you don't give 6 people the new compound all at the same time. You give one the compound and you wait like an hour and they haven't fallen over dead. So you give the second the compound and you wait another hour and the 2 of them are still alive. And then you feel comfortable dosing the rest of the people. But you kind of sit there going, are we going to kill anybody? Are we going to have an unexpected outcome? And part of that is because the way we screened, we don't, and we have a feeling like we should check certain things. And there are certain safety studies where you do things like, well, you don't shut down this pathway with this, and you test that in a test tube, and you do certain things like that. But ultimately, you're looking to see if single doses are tolerated in humans without making them sick, if multiple doses are tolerated. And once you've taken people who, and I hate to say it, but people participating in phase 1 trials are typically either students or people who really, really need the money, who participate in phase 1 studies because they can be locked up for several days in a clinical testing laboratory where you do this work and you control their diet and you draw their blood and you watch for outcomes, and they take the pill because you put it in their mouth and you make them open their mouth, and then you give them water and you make them swallow and you open your mouth again. Once you've done that, you then identify a group of people who have the disease and you start seeing, can we give them a dose that's been proven to be safe and is it efficacious? And then when it finishes, and that's phase 2, like phase 1 is proving it's tolerated in humans. Phase 2 is proving that it works in people with the disease. And then phase 3 is moving to the real world trials. And, we've just been through that and with the COVID vaccine. But once you finish your phase 3 trials and you have a preponderance of information that tells the FDA it's safe, then they approve it. And phase 4 trials are down the road. Like the observation studies you do is you start giving it to more and more people. But one of the things that comes out is, a drug like a statin drug gets labeled as it's an anti-cholesterol drug. It lowers cholesterol. Yeah. It doesn't get an indication for treating heart disease because you didn't prove it. It doesn't get an indication for being an anti-inflammatory because you didn't prove it. And it gets to be an FDA indication for a specific use, but then you and I can use it off label because we've got a couple initials behind her name that says medical doctor.
Yeah, it's true. And so the other interesting thing is when in a classic is it's approved, a chemical is approved for one indication by the FDA. It's called a drug. And then when it comes into another indication, the same chemical, sometimes they have to give it a different name, right? And the classic is Wellbutrin is an antidepressant, and the exact same molecule, Zyban, is for smoking cessation.
Right.
And so thus, it takes a lot of nuance out of how we use different things.
Right.
It takes nuance of how we use natural vitamins and minerals that affect so many different pathways. And we are slowly moving in, our lens is beginning to change that we look through in healthcare and in well-being care. And now we're talking about how does whatever we use to intervene affect function, which can affect more than just have more than one indication. So it's the world that we spend most of our time in.
Yeah. So what's the number one drug for preterm labor at this point? Is it still nifedipine after magnesium?
Nifedipine is still used to stop contractions. Magnesium is still used to stop contractions. So I'd say those are the most widely approved. When it used to be in the 50s and early 60s, IV alcohol. So I say we've improved in our use of magnesium and nifedipine.
Right. So nifedipine, if you read the package insert, its approval is as an antihypertensive drug.
Yeah.
And on contraindications, it actually labels it as not for use in pregnant women because it reduces uterine contractility.
Yeah.
So, and it has never received an FDA indication for treating preterm labor, but it is one of the primary drugs for treating preterm labor. And this is the kind of one of the most dramatic off-label pieces, where what really brought this to my attention today was having a discussion with a patient about neuropsychiatric drugs, neuroactive drugs, and how mood stabilizers, antidepressants, sleep, anti-anxiety, anti-seizure, pain, all these things, they don't hit these targets cleanly, right? They may hit a histamine receptor. They may hit an acetylcholine receptor. They may hit a norepinephrine receptor. They may hit a serotonin synthesis or reuptake receptor, and they may hit a calcium channel. They may hit an MMDA receptor. Whatever it may be, and when it does all of these things, we end up labeling things, but we don't end up knowing exactly what's going on, right? Like, oh, well, we do this for its neuropsychiatric impact.
No, that's true. And thus, Joe, we've talked about how do we make things more personalized, precise, preventative. I mean, that's part of the goal in the new era of genomics and epigenomics and recognizing this. And, it's with the metabolism of the medications it's the gene metabolism of the medications for depression and anxiety that really brings this forward. Because if you're a fast metabolizer of something, it may not work at the dosing. If you're a slow metabolizer of something, you may have more side effects. And so what are the combination of things that need to be given to affect process? And sometimes it's not another medication, although often if you give magnesium along with certain other medications, it has better efficacy, or methylfolate. But it can be indeed if you encourage exercise or different foodstuffs when you're taking the medication, it has better efficacy and less side effects. And we we are seeing that kind of more precise, personalized interventions being part of our therapeutic modalities in folks. It's just not one pill for every ill. It is, what is the train? How can we change the train so patients' function improve?
Absolutely. And the drawback to all of this is, and I think our politicians really have a lot to do with this, we do not have um, robust drug discovery as part of our national kind of emphasis on research. NIH does a lot of basic science. The applications are done by drug companies, partly because our um, our politicians don't want to charge us the taxes to pay for the expensive programs.
Yeah.
But B, I also think it's because the drug companies are paying the politicians not to make these government-owned things. And even when it is, like the research that went into, um, um, you know, the um, the mRNA technology that went into bringing us the vaccine for COVID, the research was government financed, and the patents are actually held by the government, but we're leasing them to the pharmaceutical industry at very low cost. All they had to do was manufacture it, and we actually indemnified them against lawsuits against the complications of the vaccines, as long as they manufactured them correctly.
That single fact, for anybody who's listening to this, should cause them to sit down, take a long, deep breath, and just say, wow, because it helps shine light on so different many levels of kind of an interesting business government incestuousness that, the businesses don't want to be held liable for complications. And in this country, since we lost so many vaccine makers, the only way we get vaccination makers back is to make them, is to really not allow them to suffer the consequences of badness. And there is some of the great frustration with the clear-eyed sharing of information and versus the hidden fine print of how we operate.
Yeah.
Maybe I should step off my soapbox a little bit.
Well, no, just or step aside, because I was going to talk about how, two things. One, to be fair, a lot of us in our 401ks have stock in pharmaceutical companies and insurance companies. So talking about them as if it's not us, we need to remember, and it's served a lot of us well not to pay a lot of taxes. So yes, there are people and the CEOs who are making ungodly sums of money, and the investors who are making ungodly sums of money, and the politicians who get their re-election campaigns funded have perhaps more to answer for. But we are all part of this problem, so we need to be all part of the solution. But beyond what we've talked about here is the government and our, whether the politicians, whether the administration, that's under the president, i.e. the CDC and the NIH, which is under his direction, these organizations have put forth very few solutions during COVID for other than the vaccine. And, though I do not think that there's enough evidence at the moment to support, using many of the commonly bandied about medications, including ivermectin. It is mind-boggling to me that a study in Israel was done with 90 people who took ivermectin in the setting of a residential. You're symptomatic mildly, you're being isolated and quarantined, and we're going to give you 3 days of ivermectin and see what happens to your shedding of the virus. And it seemed to work. And it was 90 people, 45 who got ivermectin, 45 who didn't. And we have a study that, like if we were in normal days, and if you and I were writing a research paper about that, in our conclusion section, we would say, this justifies doing a bigger study. Well, as far as I'm aware, more than 90 people in the world have had COVID. So it seems like there are a lot of people who could have been participants in these studies, and not just that study design. We could be brainstorming and thinking of every possible study design. Prophylaxis. We could be thinking treatment. We could be thinking inpatient treatments. We could be thinking combination studies. We have had the people to do this, but we haven't done it because, as you pointed out earlier, a drug like ivermectin has no ability to patent it. And so no one's going to be able to be the primary winner financially. And the government has refused to step forward and have a discussion about what to do.
This is a great conversation. My coffee cup is empty. It may be time to go fill it after this conversation with something different than coffee. And thanks, Joe.
Yeah. Michael's concerned that I'll say something bad about Fauci.
No,
he's trying to get me out of here before I go too far. But the final thing I'll say is we really have had a failure of leadership. We've talked about that in the last couple of weeks. People need to be leaders. People need to step forward and engage each other in conversations. And we really need, we need not to make this about belief, but about where we can meet each other in the middle.
That's right.
And, NIH, CDC, there's still going to be lots of opportunities for you guys to step forward. And there's a lot of people interested in having frank discussions about what works. I'm not an ivermectin or a hydroxychloroquine or an azithromycin advocate. I am a vaccine advocate. And I think we have the opportunity to have conversations about what works and what doesn't work before we lose more people. And they need to step forward. We need to ask our politicians to step forward. I started this out before you came on, Michael, saying, let's hear from people about what they're doing in the world to make it a better place. Whether it's simple like saying hello to somebody at a restaurant, whether it's tipping the staff in a restaurant well, whether it's saying hello to the checker, whether it's going to a volunteer place, we all need to step up and make the world a better place. So thank you. And thanks for the comments from the people who've just shared. And we're out of here for today, and we'll see you back next week.
Yeah. See you next Monday. Have a great week.
Bye-bye.
Bye-bye.